Hi everyone, today what we're going to do is we're going to talk about linkage mapping and linked genes. So with linkage mapping, we're going to kind of think about what that means, which is basically when you take a look at a chromosome, what you're trying to do is try to figure out where and how the genes are arranged and where they are located compared to each other. Now, this is not easy to do, so what we've kind of done was try to figure that out by taking a look at prophase 1 and the frequency of things crossing over. So if you want to kind of think back to the Sordaria lab with the spores and looking at the tan versus the black ones and how many recombinant ones we had, we're kind of bringing that back up again as well. But when you think about inheritance of traits, taking a look at what Mendel kind of had to say was when you take a look at, say, capital A, lowercase a, capital B, lowercase b, if they were unlinked, what Mendel says is they will independently assort and recombine with each other. So what you're going to find is essentially, right, we FOIL. We take the first A and combine it with the B to get your first gamete, your first A with the second B to get your second gamete, then you have your second A with the first B, and then second A with the second B. So what you're seeing here is, right, it's the whole idea of independent assortment. You're going to have one of each letter, one A and one B, and there's all these different possible combinations that can be created out of those four letters. Now, with linked genes, on the other hand, what we're seeing here is that the location of where these genes are compared to each other are very close in proximity. And so, therefore, you're not going to get all of this kind of independent assortment in all the four different versions. But what you might find is that because they're in close proximity, maybe one version is going to be related to the other version. And so you're not going to find as much independent assortment. You're going to have, say, the capital A always linked with the capital B and a lowercase a linked with the lowercase b, and they're kind of inherited together. So let's kind of take a look at how we determine if something is linked or unlinked. If you take a look at a test cross. A test cross is what helps you determine linkage. So by definition, a test cross is taking a heterozygous individual, crossing it with a homozygous recessive individual. So a heterozygous individual crossed with a homozygous recessive individual. That is always going to be the definition of a test cross. Now, if the genes are not linked and you were to do a dihybrid Punnett square, what you would find is that you would see this 1-to-1-to-1 ratio of the different phenotypes possibilities, okay? So a 1-to-1-to-1 ratio of a phenotype possibility. So you can see that here in this Punnett square, right? This is a test cross. You're taking a heterozygous individual crossed with the homozygous recessive. As you can see, you've got these four offspring that are going to be dominant for A and B. You have these four sets of offsprings up this column that is going to be dominant for A and recessive for B. You have another set of phenotypes here, and then your fourth phenotype here. So the ratio then for dominant, 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 recessive, recessive, dominant, and recessive, recessive is a 1-to-1-to-1-to-1 ratio. This is what you expect if there is independent assortment, right? Now, if that's not the case, what you're going to see is that things are going to be a little bit different. So what you can do is you can always test and see in a test cross if things are linked or not, if the genes are linked or not, by, again, doing this test cross, taking individuals that are homozygous, recessive, crossing it with heterozygous organisms. Now, when you take a look at the offspring, again, you're doing the same test cross that you would normally do with the other option that we just showed. But what you're going to find is that the numbers are not going to equal a 1-to-1-to-1 ratio. So kind of showing you here, just taking a look, this is not your 1-to-1-to-1-to-1 ratio. So, therefore, we're going to assume it's linked. But let's go back to the previous slide. So, parental type, what that means is that they're going to be the same as the parent. They look the same as the parent, and they're always going to be the most in numbers. But, as we see, if there's a crossover event, right, what you're going to find is that there are going to be differences than in the original parents, and those values are always going to be less. Now, with the crossing over event, because the genes are in close proximity, then they're not going to have that 1-to-1-to-1 ratio. Then you can say, okay, well, looking at the numbers, it's not a 1-to-1-to-1 ratio. It's linked, and therefore you can determine which ones are the parental types and which are the recombinant types by taking a look at this. So, coming to this example, what we're going to see here is you took parental generation, P generation, you're having purebred purple and purebred tall crossed with the purebred white and short, okay? Then what you're going to do is you're going to take the F1 test cross. So we're not making the F2 generation as in crossing two F1s, but we're doing an F1 test cross where we're taking heterozygous individual here, heterozygous, crossed with a homozygous recessive. Okay, so that's what this test cross is showing. Then in the F2 generation, the next generation from there, you can see that we do not have that 1-to-1-to-1-to-1 ratio. So therefore, we have to say that in regards to color and height, they most likely are linked. Okay, so if they are linked, then we can kind of continue on from that point, which is really kind of cool. So what you're seeing here, which you've seen this formula once before when we did the Olive Lab with meiosis. But what you can do is determine the distance, the relative distance between those two genes, height and color, by taking a look at the recombination frequency. So these formulas are actually not on your appendix. You do need to be comfortable with them because you do need to kind of be able to figure out map units. So in this particular case, to figure out recombination frequency or the percentage of recombinant frequency, you're going to take the number of recombinant offspring and divide it by the total number of offspring multiplied by 100. Then that percentage is almost like a 1-to-1 ratio. So with the idea of chromosomes, there's no real measurement in regards to length. So we just call them map units, or you might also hear them as centimorgans. Now, something to keep in mind, right, is if the genes, where they are located compared to each other, the farther apart they are, the higher the probability of them crossing over. But just remember the maximum value frequency is about 50% because you're taking a look at that distance on the chromosome. Okay, so let's do some practice problems. You will need your calculator if you don't have that out right now. But what you're going to see here is we're going to say that a test cross has already occurred, and here's the data for your F2 offspring. We're trying to figure out if these two traits, color versus height, if they are linked or not. So color and height. So for starters, what we can see is that you do not have in the offspring this 1-to-1-to-1 ratio. Again, this is after a test cross. And so therefore, we have to say it is absolutely linked. Now, from here, what we can see is we can try to determine the map units between that color and height. So if you kind of go through and you total up your values, so you'll have to use your calculator for this, you are going to see that you have a total of 1,000 offspring. Now, the next step is you want to see what the percent recombination frequency is going to be. So remember, percent recombination equals the number of recombinant offspring divided by the total. Well, what we can see and identify is that we're going to see these two are the parental type, right? We know that they're parental because they're one, most in numbers, but secondly, they are also representative of the original parents, which we're going to say is the P generation from here. So purple and tall, and then your white and short. So those are your original parental generation here. So we're going to say that those are your parental, and what you're going to see is purple and short and white and tall are different. So these are your recombinant offspring. So to figure out percent recombination, if you look at a formula, it's the number of recombinant offspring divided by total. So in this particular case, 47 plus 53 divided by 1,000. And you're still going to do percentage, so we're going to still multiply it by 100%. So stick that into your calculator. You're going to see you have 100 divided by 1,000 multiplied by 100 gives you 10%. So what this is saying is that the distance between color and height is going to be 10 map units, or you can say 10 centimorgans. And that is it. That is how you determine how the distance is in regards to map units of linked genes. Now, we can keep going further with this, and what you're going to see here is you could also determine gene order. I've got these 3 genes, A, B, and C, and I want to know how they are relative to each other on a chromosome. So, as we can see, if you take a minute, it's kind of like a little biology Sudoku. You can see 10 map units is the farthest in regards to distance, so we know that A and B are going to be the farthest apart from each other, and C is most likely going to be in the middle. But if you also notice, between B and C, it's 7 map units versus A and C is 3 map units. So you can figure out C is going to be more closely located towards A than it is to B. So this would be 3 map units, this would be 7 map units, and it gives you a total of 10. So that's how you take a look at linked genes. Now, in your packet, there are tons of practice problems. I will tell you right now, these practice problems are going to be super hard, especially some of the brain teaser ones with mapping everything out. Try your best. I know that this is actually harder than what you would ever see on an AP exam, but that's my job to push you. Again, I think Mr. Schuster may pop in. My answer key is also posted on Schoology, so you'll see that. I know that some of the links are being a little tricky, so I'm going to try to modify that and fix that all for you. But if you have any questions, please feel free to email me. Again, I hope to see you all soon, and I'll tell you more as soon as I know anything on my end. Thank you. Bye.